Hello friends, welcome to Department of Plant Pathology, TDSP Agriculture College Nasik. In previous lecture, we have studied Alternaria leaf blight of mustard, its causal organism, symptoms and their management. Now today, we are going to see the next disease, that is sclerotial wilt or collar rot or sclerotinia stem rot of mustard. Causal organism responsible for sclerotial wilt or sclerotinia stem rot of mustard is Sclerotium ralphsi and Sclerotinia sclerotiarum. This disease was first time reported in India in 1973. Now we will see the symptoms of this Sclerotinia stem rot. Initial symptoms of the disease appears 40 days after sowing. Infected plants can be spotted from a distance by their sickly appearance and in later phases the entire plant withers and dies. White cottony mycelium or white cottony mycelial growth can be seen on stem, as you can see in this image on the right side. Similarly, mustard seed sized sclerotial bodies were also formed on this white cottony mycelial growth and can be easily identified that there is an infection of this Sclerotium ralphsi. These are formed on the affected stem near to the soil level. This pathogen produces dark brown to black color sclerotia. These are hard distinct structure produced by this fungal pathogen and they can survive up to several years inside the soil. This survives in the form of infected plant debris, implements. So this secondary spread is always through sclerotia, and this windborne spores or the secondary spread of the disease occurs through sclerotia by infected tools and implements and by irrigation water. So this is all about symptomatology of this disease. The next important point is management of this sclerotial wilt. How to manage such soil-borne disease? So for that, we have to collect and destruct infected plant debris, as we have seen that infected plant debris containing sclerotial bodies, and this sclerotia can survive up to several years inside the soil. So up to the next crop, they can hibernate inside the soil, and after availability of congenital conditions, they can activate or they can infect the new crops. So first point is collection and destruction of such infected plant debris and burn it outside the field. Second most important point, it should not be forget that seed treatment with Captain or carboxine at the rate of 0.3% is very effective for controlling the initial inoculum or to avoid the initial infection of this sclerotial wilt. Similarly, this is one of the fungal pathogen, and this pathogen present near the base of the crops, so that we have to drench such bases of the plant with chestnut compounds or any another fungicidal solution at the rate of 2 ml per liter, or you can also use chestnut compound at the rate of 0.3%, which is found very effective to kill the mycelial network of fungus near the rhizosphere soil. In addition, we can also use soil amendments like goat straw, finely grounded castors and neem cakes. These are somewhat how reduces the chances of inoculum, or it reduces the disease incidence. Similarly, for such fungal pathogens, we can also use some antagonistic biocontrol agents like Trichoderma viridi and Trichoderma herzianum, which were found very effective to control such pathogen in eco-friendly manner. And such antagonistic fungi should be mixed with cow dung or should be mixed with farmyard manure at before the sowing, so that they can multiply into the cow dung or FYM, and when we apply such mixed culture inside the soil, they can easily control these fungal pathogens in eco-friendly manner. Now another most important disease is downy mildew, and its causal organism is Peronospora parasitica. Symptoms of this downy mildew belongs to the symptoms which we have studied earlier in earlier crop, that is sunflower. That means sunflower downy mildew and this mustard downy mildew are both same in symptoms, only the pathogen is different. That is, in sunflower there was Plasmopara health study, and in mustard it is Peronospora parasitica. Remaining all symptoms are same, that is yellowish color growth was seen on lower surface of leaf, hypophilous growth. These are some common symptoms of this downy mildew pathogen that are same. But we will take a quick overlook over all the symptoms of this downy mildew pathogen again. Various kinds of symptoms were produced by this pathogen, like damping off, systemic infection, local lesions, and basal rot or stem galls. In systemic infection, plants are severely stunted. Fluorosis is being seen. Fluorosis starts through midribs, causing ultimately abnormally thick, downward curled leaves that shows prominent yellow and green epiphyllous mottling. These are some particular marks of identification of this downy mildew pathogen. Hypophilous downy growth of the fungus are developed on underside of leaves, so that's why it is called as hypophilous downy growth. Flower heads of affected plants remain sterile. So this is very important point, that such downy mildew infected plants should be, or after the infection, such plants shows sterility. They can't produce seeds, so you can judge the severity of this disease. Local foliar lesions like symptoms were also shown by this pathogen and are characterized by small angular greenish yellow spots on leaves. Development of basal galls also found independently of this infection that results in systemic infection. In infected plants, flower heads are erect. So this is another particular mark of identification to identify this downy mildew. So you can easily know the primary infection of this disease, as we have seen. The primary infection of this downy mildew occurs always through soil-borne oospores. As we have seen, oospore comes under Oomycota, and this Oomycetes fungi produces oospore, and which were serves as a resting spores also, and primary source is always through, and secondary spread of this disease through wind-borne sporangia and zoospore. So this is all about disease cycle, or we can say the primary and secondary infection of this disease. Now we will see management. For managing this downy mildew, we have to use regulatory measures to prevent recess of pathogen into the India. So we can avoid the infection of this disease to new area. Similarly, we can also follow proper spacing. Dense plantation is responsible for development of this disease. So to maintain proper spacing, you have to use 60 by 30 centimeter or 45 by 30 centimeter spacing, which is optimum and recommended, so that we can avoid the incidence of this disease. Similarly, we have to root out all infected plants and should be collected and destroyed outside the field. Cropping sequence of sunflower followed by groundnut reduces the initial inoculum of this disease. Seed treatment with metalaxyl is found very effective for controlling this disease. Similarly, we can also use the eradicative measures of this metalaxyl as 2 gram 2 ml per liter of water, so that we can control this disease after the infection. So that's all for the diseases of mustard. In next lecture we will start the next topic, that is diseases of chickpea and linseed and their management. Thanks for watching this video.